Hello students, welcome to the e-learning session. Title of the e-learning unit is Free Living Nitrogen Fixing Microbes. Module name Azotobacter as Biofertilizer. I am Harshla Garg, Assistant Professor, St. Xavier's College, Mapsa, Goa. Outline of this module: identification of Azotobacter, isolation of Azotobacter, mass multiplication of Azotobacter, production of carrier-based inoculants, techniques of field application, and crop response. The learning outcome of this module is to identify Azotobacter. We'll learn the methods of isolation, mass multiplication of Azotobacter, methodology of producing carrier-based inoculants, techniques of field application, and crop response. Azotobacter is a genus of usually motile, oval or spherical aerobic free-living nitrogen-fixing bacterium. It resides in soil and rhizosphere and fixes nitrogen in association with hosts. Azotobacter includes 6 species: Azotobacter virginicae, Azotobacter paspale, Azotobacter crucocum, Azotobacter haplophilus, Azotobacter winelandi, and Azotobacter miscellus. Azotobacter is a gram-negative, rod-shaped aerobic bacterium. The cell dimension varies from 2 to 7 into 1 to 2.7 micrometer in size, and shape of each cell vary with the type of species. Cell shows varying morphology. Each cell consists of peritrichous flagella. One of the unique features of Azotobacter is that it forms an insoluble black-brown pigment containing melanin due to oxidation by enzyme tyrosinase. The optimum environmental conditions for its occurrence are: it can thrive in a temperature range of 25 to 30 degrees Celsius. It can tolerate high humidity. It requires aeration and can occur in pH range of 7.2 to 7.6 and can also tolerate high salt concentration. In order to isolate Azotobacter, one needs species of Azotobacter which can be isolated by soil dilution plate technique. 10 gram of rhizosphere soil is mixed well with 100 ml of distilled water and left undisturbed for some time to have clear suspension. The suspension is serially diluted and inoculated into petri dishes on nitrogen-free mannitol agar or Jensen's medium and incubated for 48 hours at 30 degrees Celsius. After 3 days of incubation, one can observe flat, soft, milky and mucoid colonies of Azotobacter grown on the surface of the medium. Once pure culture of Azotobacter is obtained, one can proceed for mass multiplication of Azotobacter. Azotobacter is transferred to a flask containing sterile Jensen's medium and incubated on a rotary shaker or batch fermenter for 4 days at 30 degrees Celsius. The pure cells of Azotobacter developed in broth can act as a starter culture. In 1 liter of starter culture, about 100 liters of medium is transferred to sterilized Jensen's medium in a bioreactor at 30 degrees Celsius with continuous agitation for aeration. When inoculum density reaches to 108 to 109 cells per ml of broth, it should be harvested to prepare carrier-based inoculant. Once the mass culturing of Azotobacter is obtained, one can proceed for production of carrier-based inoculants. For production of carrier-based inoculants, suitable carrier such as peat, charcoal, farmyard manure is dried and powdered passing through sieve. These carriers can be used alone or in combination. Calcium carbonate powder is added to the carrier to neutralize and it is sterilized in an autoclave. The harvested broth containing Azotobacter is poured over the carrier in such a way that 40% moisture is maintained. The inoculant is mixed and curing is done for a week. The carrier-based inoculant is packed in polythene bag and stored for further use. Coming to the application of Azotobacter inoculants in field. Azotobacter inoculant can be applied in field or to crops in following methods: seed treatment, seedling treatment, pouring of slurry or top dressing. Coming to the first one, that is seed treatment. Slurry of Azotobacter inoculant is prepared by mixing with water. Seeds to be sown in field are soaked in slurry overnight and then seeds are sown in field in the morning. The remaining slurry is directly sprayed in the field. Seedling treatment. This method is applicable for transplantation crop. Roots of seedling to be transplanted are kept in slurry of carrier-based inoculant for 10 to 30 minutes. Seedlings are transplanted immediately. Pouring of slurry. Carrier-based inoculant is diluted in water. Small amount of slurry is poured near the root zones. Slurry may also be mixed with farmyard manure and administered near root zone. Top dressing. Carrier-based inoculant is diluted in water at the rate of 2 kg per hectare and mixed with farmyard manure at 20 to 25 kg per hectare. After transplantation of cereal crops such as rice and wheat, it is broadcasted by top dressing. Crop response after field application of Azotobacter inoculant. At the Indian Agricultural Research Institute in New Delhi, field trials were conducted on different types of crop plants such as maize, sorghum, cotton, vegetable crops, wheat, rice, etc. at different places, which resulted in better response of crop plant to Azotobacter inoculant. The beneficial effect of Azotobacter is due to nitrogen fixation and also synthesis of growth promoting substances. Azotobacter is also reported to synthesize some antagonistic substances which suppresses the growth of Fusarium, Aspergillus and Alternaria, and it reduces crop diseases caused by these soil-borne pathogens. A significant increase is observed in dry matter of sorghum and maize with combined treatment of Azotobacter crucoccum and Azospirillum praesilens. These are the references for further reading. Thank you.